At the simplest level, cancer or cancer cells are cells that have lost the ability to follow the normal control that the body exerts on all cells. In our body, we have billions and billions of cells, and they have different functions. It's a very complicated process under incredibly phenomenal control. And if something goes wrong and that control is lost, and particular cells escape the normal control mechanisms, and they continue to grow, and they may spread, that's what we call cancer.
A fetus develops in the womb. It's an astonishing 40-week journey from a single cell to a baby ready to be born. Its body is a miracle of microscopic design. Tiny, perfectly formed organs, each made up of billions of perfectly functioning cells. These cells are the building blocks of our bodies. They make us what we are, 100,000 billion cells, all working in harmony.
An important point about cells is that no matter what their job is in the body, they all have the same general structure. The cells that make up our lungs, heart, or brain are all similar in their overall appearance, even though their jobs are quite different. Just as the organs that they form are made of smaller structures, cells themselves are composed of smaller parts that help them perform their jobs. These smaller structures are called organelles. Of particular importance in cancer is the organelle known as the nucleus. The nucleus can be thought of as the brains of a cell. It contains the information that acts as the blueprint for each and every one of us, just as a manual would contain instructions to assemble a chair. Specifically, this information is contained within the chromosomes that reside in the nucleus. Individual units of information are called genes. At a chemical level, genes are made of deoxyribonucleic acid, or DNA. All of our cells contain the same set of information. It is only how the information is used that makes them different. For example, cells in the lungs use different bits of the blueprint to do their job than the cells in our stomach. In cancer, changes to the DNA cause some of the genes to fail to perform or to do their job in a way that causes problems for the affected individual. In short, all cancers are thought to result from changes to DNA that alter critical genes and change the behavior of the affected cells. Cancer is the result of a long process that begins when one of the cells in an organ or tissue becomes damaged or altered in a way that causes it to break free from the normal controls that allow our cells to work together in harmony. A group of misbehaving cells can cause the same kinds of problems in a body that a defective part would cause in any other type of machine.
When the body needs it, our cells split into two and replace those cells that are defective or have come to the end of their lifespan. This makes it possible for our tissues to preserve their shape and their respective functions with the passage of time. Every cell is therefore programmed to multiply and die. This ordered but complex program is controlled by the center of the cell, the nucleus, which contains chromosomes containing many genes made up of DNA. Sometimes, some of these genes undergo a change. The nucleus then sends out abnormal orders and the cell goes wrong. It multiplies uncontrollably and takes on a life of its own. Each new cell produced contains the same defect. The cells proliferate chaotically and form a tumor. This process may be short, but is often long. 10 to 30 years may separate the birth of a first abnormal cell from the appearance of a tumor of about 1 cubic centimeter.
A normal cell will divide only when it receives a chemical signal telling it to do so. These signals are interpreted in the nucleus, and the cells reproduce their genetic information and divide into two identical daughter cells through a process called mitosis. Cancer cells do not obey this rule and will divide even if they do not receive appropriate signals. In addition to the signals that normal cells receive telling them to divide, they are also told when to stop dividing. This prevents too many cells from being made. In fact, the cell division process is a highly ordered process. This is a critical issue in cancer because cancer cells do not obey or require normal signals for division. This can lead to the formation of a mass of cells that piles up and may form a tumor. Also different from normal cells is the ability of cancer cells to continue dividing indefinitely. Certain genes make products that lead cells to reproduce. This would be equivalent to the gasoline system in cars. The genes that are responsible for making cells divide are known as proto-oncogenes. Changes in these normal genes lead to the production of oncogenes. The result may be cells that divide in the absence of proper signals. It is the equivalent of a gas pedal that is stuck in the on position, making a car go even when no foot is pushing down. Genes whose products function as the equivalent of cellular brakes also exist. As a group, these genes are known as tumor suppressors. Humans have 2 copies of each gene, one inherited from each parent. If a single copy of a tumor suppressor is damaged, the other copy is usually able to stop the cell from behaving abnormally. This would be like losing either the front or rear brakes of a car. The car may be damaged, but would still be able to stop. But if the second set of brakes is damaged as well, the car would not be able to stop, just as the cell would not be able to stop dividing if both copies of the tumor suppressor genes are damaged. One way that the development of cancer is prevented is via the death of defective cells. A cell that becomes mutated or damaged will first attempt to repair the damage. If that is not possible, the cells will commit the cellular version of suicide. An orderly process called apoptosis leads to the breakdown of key cell parts and the death of the cell. Cancer cells lose this critical capability and will continue to divide. This can lead to the accumulation of cells that can become more and more abnormal.
Cells of the immune system are capable of detecting and killing these abnormal cells, but in cases of serious disease, the rate of cancer cell division surpasses the effect of the immune system, and the tumor continues to grow.
If a change occurs to the nucleotide sequence, it is like having the letters of a word changed. An alteration in a gene is called a mutation.
Why does a cell become cancerous? There are many factors, and they can exist to different degrees. Factors have been identified, such as hereditary genetic anomalies, exposure to some viruses, HIV, hepatitis B, C, and D, papillomavirus, Epstein-Barr virus, exposure to toxic agents, chemical products, radiation, sun, unhealthy behaviors, such as the consumption of alcohol and tobacco, or a diet too rich in fat and low in fruit and vegetables. The tumor forms many blood vessels in order to survive. These will supply the tumor with oxygen and nutriments, allowing it to live and grow. This is what we call the phenomenon of angiogenesis. The tumor, however, only really becomes dangerous when the cancerous cells begin to invade through the vessels into the adjacent areas and spread to the surrounding organs. These cells can then invade other parts of the body, multiply and produce new tumors. Metastasis is the term used for this spreading process. Today, over 25 million people in the world live with cancer. 7 million die from it each year. The disease represents the leading cause of mortality for those under 65. Lung cancer is the most widespread, followed by breast cancer, colorectal, stomach, and prostate cancer. The means to combat cancer do, however, exist. These often complementary treatments are used alone or in combination according to the type of cancer and its status. The goal of the treatments is to make it possible to eliminate the tumor and cure the patient in early-stage cancer, or like a chronic disease, to control its development. There are mainly 3 kinds of treatment. Surgery consists of removing the tumor in part or in its entirety. Radiotherapy exposes the tumor to radioactive rays, which prevent the diseased cells from multiplying and destroys them. Chemotherapy consists of administering systemic medicinal substances to destroy the cancerous cells or prevent them from spreading. Chemotherapy and radiotherapy act on cancerous cells, but also on healthy cells, which explains their side effects. But today, better knowledge of the characteristics of cancerous cells makes it possible to develop therapies that target the cancer development mechanisms more specifically.